once you have a basic understanding of how hearing physiology works or auditory physiology works, understanding balance is pretty similar because inside of the structures that are responsible for helping with balance, right? Your semicircular canals, the utricle, the saccule, which we talked about are in the inner ear, there are hair cells, and those hair cells have the stereocilia associated with them, but they also have a long cytoplasmic extension known as a kinocilium. That's like a super long stereocilium. And a bunch of hair cells are actually grouped together inside of the utricle, the saccule, and the semicircular canals, like so, and they are covered by a membranous structure, like so, known as a cupula. Okay? And so what happens is you have a group of neurons, and these neurons with their stereocilia and kinocilium, right, they're connected via tip links as well. And so what happens is when the cupula bends, the hair cells, the kinocilia and the stereocilia on the hair cells are going to bend, and just as in the auditory system, these are going to be connected to potassium channels, so they're going to allow the influx of potassium into the cell, and that's going to generate the action potential, right? And so inside of the semicircular canals, the utricle, the saccule, there is a fluid that has these otolithic crystals inside of them, and those crystals make the fluid more of a gelatinous-like substance. So what I'm going to do is I'm going to draw sort of just two semicircular canals, and remember that they are on three planes. Right, so I'm going to draw one like this, and I'm going to draw the other one in blue. Just so we have different colors to show the difference of them. Like so. Okay, now inside of here, imagine that this whole thing is lined by these cells that have those cupulas on them. Okay, and then it's all around. Okay, and remember, these semicircular canals are filled with a gelatinous-like substance, jello, right? Think jello. Okay, and so what happens is imagine that your semicircular canals were in this orientation that I've drawn them here, right? Gravity is going to pull on that jello, and what's going to happen is you're going to get pull here, you're going to get pull here, and you're going to get pull here, right? So what I want to do is I want to just label these sort of regions so that we can see them a little bit better when we go to move them. This is region A. I'll make this one region B. I'm going to make this region C, this region D, E over here, and F over here. So these are our, E and F are our tips. Okay, so then let's go ahead and let's move this here, and then let's go ahead and rotate this thing in that direction. So notice that we have gravity pulling and basically sending signals in A, B, and E. Now if we rotate this, let's go ahead and rotate it so that we've got this one now sort of like this. And what's going to happen is this one then will sort of rotate this way. Okay, so go ahead and try to figure out your regions, right? This is going to be A, this is now B, right? This is still F, this is E, okay? This region here is D, and then this one's C, right? But as we rotated our head, right, now we have gravity pulling on C, D, and F, right? So before it was A, B, E, now it's C, D, F. So where gravity is pulling and where that signal is being generated is different. So with that having an X, Y, and Z axis with our semicircular canals, we can sense sort of changes in direction. So similarly with the utricle and the saccule, right? Imagine that there is fluid inside of there, and then all of a sudden, this is a gelatinous fluid. You have a plate of jello. This is your plate of jello here, right? You take off running with your plate of jello. What's going to happen with your plate of jello? Your jello is going to lean a little bit, right? It's going to lean. So when you have these hair cells, right, inside of that gelatinous fluid, you're going to cause lean in them, and then you're going to generate signals in those. So similarly, right, you get changes in acceleration. So we can now sense changes in acceleration, we can sense changes in direction, and that gives us an understanding of where our body's at in space and where it might be going in space. And so that is really cool because that will actually send signals to our brain, which will communicate with our eyes and telling our eyes where we need to look based off of where we're going, what speed we're going, all of those sorts of things, so that it can help with our balance. Right? And so this is one of the ways that you can end up with motion sickness and dizziness, right? You can disrupt the signals from your inner ear to your brain, which then communicates with your visual system, right? Or we can errantly send signals by doing things like changing the osmolarity and the gelatinous structure of that fluid. What happens if we made that jello less solid and we made it more of a liquid? It would slosh around a lot more, right? And when it would slosh around right more, move back and forth, right? That's going to tell our body, hey, we're moving, we're moving, we're moving, right? And you're going to get all these signals that you're moving a lot when you're just barely moving, and that's going to cause you to be motion sick. So enhancing or upregulating the signals inside of your inner ear can lead to things like motion sickness and dizziness.